A tissue with high population of lymphocytes and macrophages is called lymphoid tissue. Anatomical entities that are significantly made up of lymphoid tissue are called lymphoid organs. Thymus and bone marrow are primary lymphoid organs where T lymphocytes and B lymphocytes mature respectively. T cells are involved in cell-mediated immunity. They are of two types, helper T cells and cytotoxic T cells. They differ in cell surface proteins or receptors. B cells are responsible for humoral immunity. They are differentiated into plasma cells, which in turn produces antibodies. Both T cells and B cells are richly populated in secondary lymphoid organs. They are spleen, lymph nodes, and mucosa-associated lymphoid tissue, tonsils, appendix, and Peyer's patches. Spleen filters the blood. It tackles the antigens and foreign particles present in the blood. Lymph nodes filter the lymph. If wound is contaminated, dust particle enter the tissue space. How it is taken care by the body? This shows the capillaries at the tissue level, afferent end and efferent end of the capillary. There are some blindly opening lymphatic vessels at the capillary. Not only dust particle, if any viruses, bacterial body walls, dead cells, broken cells, large molecular weight proteins, if they are within the tissue space, outside the cells, extracellular fluid is usually getting back into the venous end of the capillary. If molecular weights are more than that which cannot enter the venous end of the capillary, they will pass into the blindly opening lymphatic vessels. This is how the blind end of lymphatic vessels show flap valves which will open inside and tissue fluid is taken inside with all the foreign particles. Lymph from different parts of the body convergingly drains through different orders of lymph nodes and finally drains into the major lymphatic vessels in the thorax and to terminate into the venous system. Lymph is clear fluid without having RBCs. Lymph node is capsulated bean-shaped organ having many afferents and only one efferent at its hilum. Supporting structure is made up of capsule and trabeculae and also reticular fibers inside the parenchyma which hold the lymphocytes in place. Lymph passes in through the afferent vessels. It seeps below the capsule in subcapsular sinus and by the side of the trabeculae in the paratrabecular sinus, and finally through the medullary sinuses it goes out through the efferent vessel. In these sinuses, the density of lymphocytes are lesser and cells are loosely arranged to allow the lymph to pass easily. Parenchyma is divided into cortex and medulla, where lymphocytes are densely packed within the reticular fibers. It is divided into cortex and medulla. It shows lymphatic nodules in the cortex and medullary cords in the medulla. How lymphocytes enter the lymph node? Lymphocytes enter the lymph node through the blood vessels from the hilum and after the capillaries, post-capillary high endothelial venules will allow the lymphocytes to escape out the capillary into the lymph node. Supportive structure of the lymph node, capsule and trabeculae. Extension of the capsule are trabeculae. Reticular fibers are not seen in H&E. They are in mesh holding the lymphocytes inside. Parenchyma is cortex and medulla. Cortex is darkly stained with lymphocytes. Medulla is having medullary cords and sinuses. This is subcapsular space. These are paratrabecular spaces or sinuses, and these clear spaces are medullary sinuses. Cortex has lymphatic follicles or lymphatic nodules. It has primary follicles and secondary follicles. Secondary follicles have centrally pale staining germinal centers. In the medulla, we find medullary cords and medullary sinuses. This is higher power of capsule and parenchyma. Below the capsule, we can see sinus, subcapsular sinus, where cell population is sparse, which allows the lymph to pass through. This is a structure in medulla where we find medullary cords, populated cords of lymphocytes, and clear spaces are medullary sinuses. Lymphatic follicles are of two varieties, primary follicle, secondary follicle. These are also known as lymphatic nodules. Secondary follicle has pale center. This is called germinal center. This happens after antigenic stimulation. This is called activated lymphatic nodule. This shows secondary follicle having pale central zone, that is germinal center, and darker peripheral zone, that is mantle zone. In the mantle zone, resting B cells are there. In the germinal center, clonal differentiation is happening. B cells are dividing, so they have pale staining. At the junction of cortex and medulla, we find paracortex, which is richly populated with T lymphocytes. Diagrammatic representation of capsule, trabeculae, outer cortex, inner medulla, paracortex. Parenchyma shows lymphatic nodules, primary and secondary lymphatic nodules. Subcapsular sinus, paratrabecular sinus, medullary cords, medullary sinuses. This is higher magnification of the cortex, subcapsular sinus, secondary follicle. This is how all the antigens flowing through the lymph are filtered in the lymph node and are presented to lymphocytes, macrophages for immunogenic reactions. Lymph nodes drain lymph. In the same way, spleen drains blood. For the same reason, spleen is also known as hemolymphoid organ. We will see the histology of spleen in the next section.